What if living bacteria could store and process information, acting like tiny biological computers? This is the exciting concept behind neural biofilms, films of microorganisms that exhibit behaviors similar to a brain, such as adapting to changes and remembering environmental conditions. Unlike traditional memory systems in electronics, which rely on rigid, non-living structures, neural biofilms leverage the natural adaptability of bacteria. These bacteria can communicate with each other through chemical signals, adjust their behavior based on their surroundings, and even pass on these adaptations to future generations. Imagine biofilms that could monitor pollution levels in water, record health data inside the human body, or even perform computation at microscopic scales where normal computers cannot function. Their self-healing and self-replicating properties make them durable and potentially revolutionary for applications in medicine, environmental monitoring, and even futuristic technologies like biocomputers. By programming bacteria to perform specific tasks using advances in synthetic biology, researchers can create living systems capable of storing data or responding intelligently to their environment. Neural biofilms are not only a fascinating blend of biology and technology, but also a glimpse into a future where living systems work alongside machines to solve some of humanity's biggest challenges. However, this innovation comes with important questions about safety, ethics, and environmental impact, making it a topic of both immense promise and careful consideration.
Part 1. Introduction to Neural Biofilms and the Concept of Memory. Neural biofilms are films of bacteria or other microorganisms that exhibit coordinated, adaptive behaviors, potentially mimicking some aspects of neural networks. Memory, in this context, refers to the ability of a system to store and recall information over time. Recent research suggests that bacterial biofilms could be capable of memory storage, leveraging their collective behavior, communication pathways, and adaptability to environmental changes. This idea opens new frontiers in bioengineering, where biofilms might serve as biological memory devices. Example: certain biofilms can remember environmental conditions, such as exposure to antibiotics, by altering their gene expression and behavior in subsequent generations. Analogy: biofilms are like a distributed cloud storage system where individual bacteria act as nodes, collectively storing and processing information. Biofilms are communities of microorganisms embedded in a self-produced extracellular matrix. This matrix consists of polysaccharides, proteins, and DNA, creating a structured environment that facilitates communication and resource sharing. The biofilm's highly organized structure resembles a rudimentary network, allowing for coordinated responses to stimuli. The dense and interconnected nature of biofilms suggests that they could support the storage and transmission of information. Example: Pseudomonas aeruginosa biofilms create channels for nutrient flow and waste removal, showing spatial organization that could be repurposed for computational tasks. Analogy: biofilm structure is like the circuitry of a computer motherboard, where pathways and nodes facilitate the flow of information and resources. Biofilms exhibit forms of memory through their ability to adapt to environmental challenges and retain changes over time. This memory is encoded at the molecular level, often involving epigenetic modifications, protein folding, and quorum sensing, a communication system where bacteria release and respond to signaling molecules. Quorum sensing allows biofilms to process and integrate information about their environment, adjusting behaviors collectively. Example: Bacillus subtilis biofilms can remember exposure to harsh conditions, increasing resistance in future generations through heritable gene expression changes. Analogy: biofilm memory is like a chalkboard that retains marks until actively erased or rewritten. Adaptations persist unless environmental conditions necessitate change. Biofilms share several features with neural networks, including parallel processing, adaptability, and distributed information storage. Individual bacteria within a biofilm act like nodes in a network, communicating via chemical signals and modifying their behavior based on collective feedback. This decentralized system allows biofilms to learn from environmental inputs and optimize responses, similar to synaptic plasticity in neural systems. Example, in Myxococcus xanthus, biofilms demonstrate collective motility and resource allocation, reminiscent of neural coordination. Analogy: biofilms are like an ant colony where individual ants' bacteria work together to solve complex problems, mimicking neural computations. Harnessing the adaptive and memory-like properties of biofilms could revolutionize data storage technologies. Biofilms could serve as biological memory devices, capable of storing information through genetic or biochemical encoding. Such systems might be used in environments where traditional electronic storage is impractical, such as extreme conditions or microscopic scales. Biofilms' self-replicating and self-healing properties make them uniquely suited for durable, long-term memory applications. Example, engineered biofilms could store binary information by flipping gene expression states in response to specific stimuli, creating a living, rewritable storage medium. Analogy: using biofilms for memory storage is like writing on a living notebook. Its pages can rewrite themselves and heal if damaged. Despite their potential, biofilms face significant challenges as memory storage systems. Ensuring precision in information encoding, stability over time, and reproducibility across different biofilms are key hurdles. The inherent variability of biological systems and susceptibility to mutations could complicate data reliability. Additionally, interfacing biofilms with electronic systems for hybrid biocomputing presents technological and engineering challenges. Example, mutations in biofilm-forming bacteria can disrupt quorum-sensing pathways, leading to inconsistent behavior and loss of stored information. Analogy: managing biofilms for memory storage is like taming a wild garden. Without careful cultivation, it may grow unpredictably and lose its intended structure. While the evidence for non-chemical cellular communication is growing, significant challenges remain. Measuring and manipulating subtle signals like electromagnetic fields or biophotons requires advanced technologies and interdisciplinary approaches. Future research should aim to clarify the mechanisms, specificity, and biological significance of these pathways. Understanding how they integrate with traditional chemical signaling will be critical for developing a unified model of cellular communication. Example, advanced imaging techniques such as quantum-based sensors are being developed to detect weak electromagnetic fields and biophoton emissions in real time, paving the way for new discoveries. Analogy: investigating intercellular communication beyond chemical signals is like decoding a hidden layer of a complex operating system. Unlocking these secrets could reveal entirely new functionalities. Biofilm-based memory storage could have transformative applications in biotechnology and medicine. For instance, biofilms could serve as environmental sensors, recording data about pollutants or toxins. In medicine, engineered biofilms could monitor and respond to changes in the human microbiome. Storing information about disease progression or therapeutic interventions. These living memory systems could also be integrated into wearable or implantable devices for continuous monitoring. Example, biofilms engineered to detect heavy metals in water could encode contamination levels in their genetic material, providing a biological record of environmental conditions. Analogy, biofilms as sensors are like living diaries, recording events in their environment for later analysis. The use of biofilms as memory devices raises ethical and environmental concerns. Introducing engineered biofilms into natural ecosystems could have unforeseen consequences, such as outcompeting native species or disrupting ecological balances. Additionally, the potential misuse of living memory systems for surveillance or unauthorized data collection presents ethical dilemmas. Careful regulation and risk assessment will be crucial for responsible development. Example, Engineered biofilms designed for industrial use could accidentally escape into the wild, spreading engineered traits to natural bacterial populations. Analogy: deploying engineered biofilms without safeguards is like releasing an experimental drone without a navigation system. It might stray and cause unintended harm.